Welcome to part 9 of this video on the immune system. Okay, so in this video what we're going to do is we're going to start discussing the adaptive immune system. So we've now finished the innate immune system. We've discussed both parts of the innate immune system. We've discussed the first line of defense, which is the physical and chemical barriers that prevent pathogens from gaining access to our tissues, and we've discussed the second line of defense, which is the acute inflammatory response, which is the way that we can eradicate pathogens which have breached the first line of defense and which are infecting our tissues. If the second line of defense, the acute inflammatory response, fails to clear the pathogen, what we're now going to initiate is an adaptive immune response against the pathogen. And this is often also sometimes referred to as the third line of defense. Okay, so the way I want to do this is I want to start off with a little sort of briefing of what the adaptive immune system is going to be all about. I want to then talk about the fact that we can divide the adaptive immune system into two different portions, the humoral adaptive immune response and the cell-mediated adaptive immune response. And I then want to talk about what is actually going to initiate the adaptive immune response, namely antigen presentation by antigen-presenting cells. Okay, so to begin with then, let's have a reminder of the basic principles of the adaptive immune system. So remember, the innate immune system was non-specific. All of those mechanisms in the innate immune system, they were the same mechanisms that we would employ no matter what pathogen it was that was actually attacking us. The adaptive immune system is different. These mechanisms are going to be specific to the pathogen that is actually currently attacking us. We are going to produce whole armies of lymphocytes, special types of white blood cells that are specifically tailored to fight the pathogen that we are currently working with. They will attack antigens that are associated with the current pathogen that is attacking us. So this is the first concept that we need to be absolutely clear on, the concept of an antigen, because we are going to be using this term all the time from now on. So, an antigen is a protein molecule that could have lipid and carbohydrate modifications made to it, which is capable of initiating an adaptive immune response. It's capable of having an adaptive immune response initiated against it. Now, a single pathogen might have a huge number of different antigens. So if I just draw a little picture of my pathogen here, let's say this is my pathogen, so I'll just colour it in once again in red here. It might have a huge number of different proteins on its surface, which are its unique proteins. They're very different from human proteins, so they might look very different, and therefore we can initiate adaptive immune responses against these and not be at risk of attacking our own cells. So I want to highlight these in different colours to stress that there might be loads of different antigens that we can launch adaptive immune responses against. So let's have that one in green, that one in yellow, that one in blue here, we'll have one in pink, and then a final one I think in orange. So what I'm trying to stress here is that a single pathogen will have a huge number of different proteins associated with it. I've drawn loads of integral membrane proteins. Antigens don't necessarily need to be surface proteins in this way. They could be proteins that the pathogen is secreting and using as toxins to attack our human cells. So I might draw one of those here. So let's say this is some secreted protein by the pathogen. This could be an antigen as well. It could have an adaptive immune response initiated against it. The point here is that a single pathogen will have a huge number of potential targets for adaptive immune responses. It will have a huge number of different antigens, and therefore a single pathogen is not just going to get one adaptive immune response initiated against it. It's going to get a huge number of different adaptive immune responses initiated against it, against loads of its different antigens. Okay, and as we'll actually see later on, a single antigen can actually have loads of different adaptive immune responses initiated against different parts of it. So, you know, a single protein here, it might be a massive great thing with loads of different bits that look very different and have very different chemical properties. The different bits of a antigen are known as the different epitopes. So this is the concept now that an antigen might be a massive great protein. So if I just draw a bigger picture now of this pink one, let's say it looks like this, and you can see that I'm deliberately trying to show the fact that, you know, it's hugely varied. It looks very different if you look at the different portions. So it's got loads of different facets to it, and in fact all these different epitopes, as they're called, the different parts of the antigen, can have their own adaptive immune responses initiated against them. So a single pathogen might have loads of antigens, a single antigen might have loads of epitopes, and that means that we can initiate absolutely loads of different adaptive immune responses against the same pathogen. Okay, so do not get confused and think that a pathogen has one antigen and is just going to get one adaptive immune response initiated against it. A single pathogen will have loads of adaptive immune responses initiated against it. Okay, so that's the concept of an antigen. Really important that we understand that before we begin this discussion properly. The next important concept that I want to bring up now, is the concept that we can split the adaptive immune response into two different parts. Okay, so we can categorize it into two different types of response. A response which is known as the humoral adaptive immune response, and a response which is known as the cell-mediated adaptive immune response. And before we go any further, I want to explain what's the point of having these two different types of responses. What are they actually against? Well, this is the difference between attacking extracellular pathogens and intracellular pathogens. So, the humoral adaptive immune response can attack extracellular pathogens. So if I've got a pathogen that is not inside of cells at the moment, so here is my human cell, and we can see that the pathogen there is in the tissue fluid sitting alongside the human cell, and we don't like the fact that it's there, but at least it's not inside the actual cells. The humoral adaptive immune response is fantastic at getting rid of pathogens that are extracellularly. So in this case, this is the extracellular case. The humoral adaptive immune response is capable of attacking pathogens whilst they are extracellular. Okay? The cell-mediated adaptive immune response is then the response that can actually get rid of pathogens that are inside of cells. The humoral adaptive immune response, brilliant though it is not capable of getting at pathogens that are inside of cells. It can't kill them if they're inside of cells. Whereas the cell-mediated adaptive immune response, this has greater powers. It's actually capable of killing them when they're inside of cells. Or different powers, maybe I should say, rather than greater powers. So it's capable of killing pathogens that are actually inside of cells. So this is the difference. So humoral is to annihilate pathogens whilst they're extracellularly. The cell-mediated response is to annihilate pathogens if they're intracellularly. Okay, this one cannot kill extracellular pathogens. The cell-mediated response kills, this one, the humoral adaptive immune response cannot kill intracellular pathogens. The cell-mediated response kills intracellular pathogens by causing the actual host cell here, which I'll just colour in yellow, to commit apoptosis, to commit suicide, and in that process it will destroy the pathogen that's inside of it. Okay, now most pathogens will get a humoral adaptive immune response initiated against them, at least one, multiple most likely, as I've just explained, because the different antigens and the different epitopes of antigens can have different humoral adaptive immune responses initiated against them. The reason that most pathogens will get that is that most pathogens do at some point have to be extracellularly, and therefore they are targetable by a humoral adaptive immune response. Even the intracellular pathogens, generally they have to go extracellularly for a while to get from cell to cell, so they have to go from cell to cell via the tissue fluid. Occasionally it's the case that there's a way that they can get from one cell to another cell without going through the extracellular fluid, but usually they do have to be extracellular for some time, and whilst they are extracellular, they are targetable by a humoral adaptive immune response, and therefore they will get humoral adaptive immune responses initiated against them. Whereas cell-mediated adaptive immune responses, they're only going to be initiated if the pathogen spends some of its time inside your own cells. If it's purely extracellular and it can't go inside of your own cells, then you're not going to initiate cell-mediated adaptive immune responses. Okay, so I hope that that's clear, and we will continuously be splitting our discussion into the cell-mediated and humoral adaptive immune responses. Okay, so that's the beginning discussion now complete. Let's actually begin discussing the adaptive immune responses. So I want to give a warning straight away. This is a very, very complicated system, and it takes a while before you will see the big picture. So initially you're just going to have to bear with the discussion, and then hopefully by the end you will be able to see the big picture of how this all fits together. Okay, so the first part then is that we need to initiate an adaptive immune response. And even if you don't know anything about lymphocytes at the moment, you should know that lymphocytes are the key players in the adaptive immune response, and we will be discussing them in so much detail in just a moment. To initiate an adaptive immune response, we need to activate lymphocytes, and the lymphocytes are going to be directed against specific epitopes of specific antigens. Okay, so how are we actually going to activate the lymphocytes once we've got a pathogen present within our tissue? Well, it's going to be done with the help of special cells that are going to take the antigens, well, that are going to take fragments of the antigens to lymphocytes to ask lymphocytes whether they can actually attack this antigen that we've got here, and if they can, they're going to be activated. So this is the first process that we're going to discuss, the process of antigen presentation to activate lymphocytes. Okay, so this is carried out by special cells that are collectively known as antigen presenting cells, more commonly just abbreviated down to APCs. So this stands for antigen presenting cells. Okay, and we've actually met the major examples of antigen presenting cells already. They are the dendritic cells and macrophages, particularly the dendritic cells. Dendritic cells are absolutely fantastic at activating lymphocytes, at activating the adaptive immune response. Macrophages are slightly less good at doing it, but they can also do it. For the purposes of our discussion and for the purposes of drawing pictures, I'm going to assume that we're working with a macrophage, and the reason for that is simply because dendritic cells take a lot more effort to draw than macrophages. So let me just get my picture back up here. So here's my pictures from the innate immune system. Remember, here are the dendritic cells and here are the macrophages. You can appreciate that drawing this is a lot more effort than drawing this, and it's also a lot easier to show what's happening inside a macrophage than it is to show what's happening inside of a dendritic cell, because I've got much more space to work with on here. That is the reason I'm going to draw all my pictures as macrophages. In reality, the dendritic cells are far better at doing this than macrophages, and the major initiators of the adaptive immune system are the dendritic cells. So in the ideal world, I would have more effort, more guts, and I would be drawing my pictures as dendritic cells, but I'm going to draw my pictures as macrophages. But be aware that the major one doing this is dendritic cells rather than macrophages. Okay, so what's going to happen then now is that the antigen presenting cell, whether it's a macrophage or a dendritic cell, is going to present the antigens of our pathogen to lymphocytes. And the way that it presents these antigens is not as a complete antigen, it just presents the little fragments, and you'll see how this all fits together later on. But for now, just accept that it's going to present little fragments. So it's going to firstly chop up antigens into little fragments and then present the fragments to lymphocytes. And the way that it presents them is different depending on whether we are dealing with an intracellular pathogen or an extracellular pathogen. And this relates to the fact that if we're dealing with an intracellular pathogen, we're going to be initiating cell-mediated responses. If we're dealing with an extracellular pathogen, we're going to be initiating humoral adaptive immune responses. So we're already seeing the difference between these two types of adaptive immune response. So what I'm firstly going to discuss then is the mechanism of antigen presentation by these antigen presenting cells if we're dealing with an intracellular pathogen. Okay, so let's start with the intracellular case and then we'll go on to the extracellular case. So intracellular. So what we will have then is we'll have some cell, so let's say this is a human cell in our tissue, like so, which has the pathogen inside of it. Here we go. Okay, so here is the intracellular pathogen. Now, when you have the pathogen inside of you, this cell will be very obvious. It will get all sorts of pathogen-associated molecular patterns associated with it. So the dendritic cells and the macrophages can tell that this host cell is infected by the pathogen. And what they will actually do is they will phagocytose this entire cell with the pathogen inside it. And then what they're going to be able to do is present antigens from that pathogen to T cells, to lymphocytes. Okay, so for this discussion, what I'm going to now need is to have a specific antigen that we're working with. So what I'm going to draw on my pathogen here is a specific antigen here, and I've drawn it really bluntly, very bigly, very large rather. So here that green blob is going to represent an antigen on the surface of the pathogen that we are going to be initiating an adaptive immune response against. As I have already discussed, we will have loads of different antigens, and this same thing will be happening for all of those different antigens. But to make this simple, I'm just going to follow the story for one antigen and follow one adaptive immune response. But be aware that you will not just be initiating one adaptive immune response, you'll be initiating loads of them against the same pathogen. So here we have our intracellular pathogen with the antigen that we're going to initiate the adaptive immune response against. What will now happen is this is going to be phagocytosed into our antigen presenting cell, which I'm drawing as a macrophage here. So here's our great big macrophage. Here is its nucleus, and I'm now going to draw its nucleus in a more fancy way. So I'm going to draw a more fancy picture of its nucleus. So remember, the nucleus actually has a double membrane, and the space between the inner and outer membranes is continuous with the rough endoplasmic reticulum, and that's the reason I want this to be drawn now, because I want a picture of the rough endoplasmic reticulum here. So here's now my more fancy picture of the nucleus. So this is the inner nuclear membrane, and I'll just colour it in a little bit here. So in green here, this is the inner nuclear membrane, and then outside of that we've got the outer nuclear membrane, and you can see that the space in between the two membranes is continuous then with this space here, and this is going to be our rough endoplasmic reticulum, and we're going to need this for our discussion in just a moment. So little r, capital ER, stands for rough endoplasmic reticulum. And remember, the rough endoplasmic reticulum is the site of protein synthesis. It's a major site of protein synthesis. It's covered in ribosomes. It's also a major site where proteins that are going to be put on the cell membrane are going to fold, and they're going to start going into membranes in the rough endoplasmic reticulum, and then they'll be trafficked to the cell membrane in ways that we will see in just a moment. So this is the rough endoplasmic reticulum here. Okay, and of course we've phagocytosed our pathogen-infected cell. So here is our phagosome here, and then inside of the phagosome now, here is our pathogen-infected cell. I'm drawing its nucleus in just the normal, cheap way, and then we've got our pathogen inside here with the ever-important antigen there. So I'll just colour the pathogen in red here, and then there is the antigen. So I hope this picture is clear. So we've phagocytosed our infected cell here, and I'll just colour the cell membrane in yellow here, into a phagosome, and now we're going to have the possibility of taking this antigen and presenting it on our surface. So the mechanism of this isn't completely understood. So what's going to happen, of course, is the normal phagocytosis process is going to occur. The cell is going to be broken down by the lytic enzymes that are put in there by the lysosomes, and also by those incredibly reactive molecules that are put in as well, that we've now seen and understand. But what happens, amazingly, is a process that is not understood. We don't know the mechanism of this, but certain of the pathogen's antigens are transported from the phagosome into the cytoplasm of the antigen-presenting cell. So I'm just label this up as our antigen-presenting cell, and obviously this is a macrophage that I've drawn, as I've discussed already, rather than a dendritic cell. So what's actually going to happen is our antigen here is going to end up in the cytoplasm of the cell, like so. Okay, and what's then going to happen is it's going to be broken down into fragments in the cytoplasm of the cell, and the means by which it's going to be broken down into fragments is via an apparatus known as the proteasome. So I'll just draw this here. So the proteasome is something rather terrifying in the world of molecular biology. It's terrifying if you're a protein. The basic idea is that proteins go into the proteasome. It's a tube, and proteins go in one end, and fragments of proteins, little tiny little peptides, come out the other end. So it chops proteins up into loads of little bits. So this is the proteasome here in yellow, and it is just a massive great tube. So what's going to happen is the antigen that we have brought in to the cytoplasm from our phagosome here is going to go through the proteasome, and then it's going to be chopped into loads of little fragments. And it's these fragments that are now going to actually be presented on the surface of the antigen presenting cell. So what now happens is these little fragments, and again I'm just going to follow the story of one of these little fragments now, but all of the little fragments will have their own equivalent stories. So here is one of these little fragments now, drawn in orange here. What's going to happen is these little fragments are going to be transported into the rough endoplasmic reticulum by a transporter on the surface of the rough endoplasmic reticulum. So here it goes, it's being transported into the rough endoplasmic reticulum, and this transporter here is known as the TAP. And this stands for the transporter associated with antigen presentation. So the T is for transporter, and then it's associated with antigen presentation. So the T is for transporter, the A is then for antigen rather than associated. That's the more important bit here. Transporter antigen presentation is effectively what the TAP stands for, but in full its name would be the transporter associated with antigen presentation. So this is just a transporter on the surface of the rough endoplasmic reticulum that is going to transport our fragments of antigens that we now have in the cytoplasm of this cell into the rough endoplasmic reticulum, like so. So there in blue, that's the transporter associated with antigen presentation. Okay, and now what's going to happen is these fragments of antigens are going to get loaded onto a protein complex, which is then going to be the mechanism by which we display it on the surface of the on the surface of the antigen presenting cell. And this is a protein complex by the name of major histocompatibility complex class 1, and at the moment this is in the membrane of the rough endoplasmic reticulum. So I'll draw it there. So here, drawn in pink, this is the major histocompatibility complex class 1, often abbreviated down to MHC class 1. So we'll see that antigen fragments from an intracellular pathogen are going to be loaded on MHC class 1 by this mechanism, whereas antigen fragments from an extracellular pathogen are going to be loaded on a different one known as MHC class 2 by a mechanism that we'll come on to in a moment. So MHC class 1, this stands for major, that's the M, and then the H is for histocompatibility, all one massive great big word, histocompatibility, and then the C is for complex, and then it's major histocompatibility complex class 1. Okay, so that's what we're seeing there, and we will discuss the structure of this major histocompatibility complex class 1 in more detail in a moment, but at the moment I just want to give you the big picture of how the antigen fragment is going to be presented on the surface of the antigen presenting cell. So we'll come back to this because it is important that you know a little bit more about this, because it's complex, these proteins, and understanding something about these proteins is very important when we consider things like autoimmune diseases and who is at risk of having autoimmune diseases, but I'll come back to that discussion later on. So where have we got to so far? So we have our host cell that is infected with this intracellular pathogen, and to initiate a cell-mediated adaptive immune response against this intracellular pathogen, what has to happen is one of these antigen-presenting cells, and we're drawing a macrophage here as I've discussed, is going to phagocytose the infected cell, and it's going to break it down, and then by a mechanism that's not well understood, some of the antigens of the pathogen are moved from the phagosome here into the cytoplasm of the antigen-presenting cell, and then what's going to happen is they're going to be broken down by the proteasome into little fragments. And by the way, these little fragments will need to be between 8 and 10 amino acids long. So alpha stands for amino acids. So these fragments in orange here, they're going to need to be between 8 and 10 amino acids long. They're then going to be transported, and of course you'll get loads of these different fragments. They're then going to be transported from the cytoplasm into the rough endoplasmic reticulum, and some of them will then end up being presented on MHC class I proteins, which are currently in the membrane of the rough endoplasmic reticulum, and we will discuss what characteristics the fragment has to have in order to be presented on an MHC class I protein. It is not the case that any old fragment between 8 and 10 amino acids long can be presented on an MHC class I protein complex. There is a requirement, but we'll discuss that more in detail later on. For now, just know that some of the fragments associated with some of the antigens of the pathogen that we've broken down in this way will now end up bound in that little groove of the MHC class I protein, and that little groove is known as the peptide binding groove. So I've drawn a little space there, and I've put now the orange fragment in there, and that little space was known as the peptide binding groove. Okay, so now what's going to happen? Well, of course, now the MHC class I with its little fragment bound in its peptide binding groove, this complex of the antigen fragment with the MHC class I is now going to be transported from the rough endoplasmic reticulum to the plasma membrane, and it will go through the classical secretory pathway. So if I just draw this out here, so let's say this is the rough endoplasmic reticulum here, and I'll just draw the nucleus in as well, like so. So there is the outer nuclear membrane, and here's the inner nuclear membrane. So at the moment, this is what we've got. So here is the major histocompatibility complex class I. There's the antigen fragment between 8 and 10 amino acids long, here in orange, and I'll just colour this MHC class I in pink here as well, like so. So now what's going to happen is this is going to be moved from the Golgi apparatus, from the rough endoplasmic reticulum to the Golgi apparatus, and then it's going to work its way through the Golgi apparatus into a secretory vesicle, and then that secretory vesicle will fuse with the plasma membrane. So let me now draw the Golgi apparatus here. So the Golgi apparatus, remember, consists of loads of these cisternae, which are discs, membrane enclosed discs, like so, stacked on top of one another. So here's my picture of the Golgi apparatus, like so. So it usually has about 7 of these discs. I'll try and draw 7 on here. There we go. So this is representing the Golgi apparatus, and as you can see, one side of the Golgi, the large side of the Golgi, which is known as the cis Golgi, cis means on the same side, so this is the side facing the rough endoplasmic reticulum. The large side is the cis Golgi, and that's facing the rough endoplasmic reticulum, and then there's this other side, which is the smaller side, facing away from the rough endoplasmic reticulum. This is then called the trans Golgi. Trans means on the other side. Okay, right, so what's going to happen is you're going to deliver the MHC class I complex with its peptide fragment from the rough endoplasmic reticulum to the trans Golgi, to the cis Golgi firstly, and that will just be done by a little vesicle budding off the rough endoplasmic reticulum and moving to fuse with the membrane of the cis Golgi here, and what that will then result in is our MHC class I will now be in the Golgi here with its peptide fragment still there. So this is the pathway that it will follow. So here it goes up to the Golgi, in pink there, and then there's the peptide fragment in orange, and then what will happen is it will gradually move from cisternus to cisternus, and it will move through the Golgi from the cis Golgi to the trans Golgi, and it will end up in the membrane over here, and then what will happen is a vesicle will bud off from there, like so, and I know this is out of scale, but you get the message here. So here is the little secretory vesicle with our MHC class I complex here in pink, with the peptide fragment in the peptide binding groove, and I colour that in orange. And now this secretory vesicle is then going to move over to the plasma membrane. So this is the plasma membrane here. So I'll just label this up as the secretory vesicle that's come off from the trans-Golgi. So this is the secretory vesicle. This will now move to the plasma membrane here. It will fuse with the plasma membrane, and of course what will end up happening is we'll end up with our MHC class 1 complex with the peptide fragment like so. So here is the MHC class 1 in pink here, and here is our peptide fragment in the peptide binding groove of the MHC class 1. So what will overall end up happening is this antigen presenting cell will end up coated in MHC class 1 complexes with fragments of antigens from our pathogen present in their peptide binding grooves. So remember, we followed the story for 1 antigen and 1 peptide fragment. This will be happening for loads of different antigens and loads of different peptide fragments of those antigens, so you will get loads of different MHC class 1 complexes on the surface of the antigen presenting cell with loads of different peptide fragments in them that are associated with this pathogen. And this antigen presenting cell with antigen fragments now presented is then going to go off and try and activate T lymphocytes. Okay, so 2 really important terms then are the term of antigen processing and antigen presentation. So I just want to make absolutely clear what these mean. Antigen processing is the process that we've just been through. So taking the antigen, breaking it down into fragments, and then putting them on MHC class 1 complexes, that is antigen processing. Antigen presentation is then when you actually put these antigen fragments on the surface of the cell, this process that we've been through here. So we have just been through the process of antigen processing and antigen presentation by an antigen presenting cell. Okay, and we've done it so far for the case of an intracellular pathogen. So for the case of an intracellular pathogen, these peptide fragments need to be between 8 and 10 amino acids, and they're going to end up being presented on MHC class 1, and this is then going to activate a cell-mediated adaptive immune response. What I now want to go through is what's going to happen for an extracellular pathogen, because it's the same concept, a very similar concept, identical concept. Pathways are slightly different for processing and presentation, but the big difference is that we're going to have a different MHC complex. We're going to have MHC class 2, and that then is going to activate humoral adaptive immune responses rather than cell-mediated adaptive immune responses. Okay, so let's now go through the pathway for an extracellular pathogen. So let's now say that our pathogen is extracellularly rather than intracellularly. Okay, so extracellular. So this time what we're going to have again is we'll have a pathogen, and we'll follow the story for a single antigen this time again. So here is our pathogen, but this time it's extracellularly. And remember, I'll remind you that even intracellular pathogens will often have to be extracellular at some point, and therefore will be capable of initiating humoral adaptive immune responses as well as cell-mediated adaptive immune responses. So don't just think that intracellular pathogens initiate this and extracellular pathogens initiate this. Intracellular pathogens are most likely going to initiate this and this, because they will be extracellular at some point, and therefore this pathway can occur as well. Okay, so here is our extracellular pathogen. Here is its antigen here, and what's now going to happen, of course, is it's going to get phagocytosed by one of these antigen-presenting cells. So it'll be phagocytosed by a macrophage or a dendritic cell. So again, I'm going to draw my picture of the macrophage, and I'm going to draw it bigger this time, because last time our picture was quite small, so it was difficult to see on the camera. Okay, so here is our picture. So I'll have the phagosome down here. And then here is the pathogen inside the phagosome, and there is the antigen on the surface of the pathogen. Shown completely out of scale, but it is just a picture to get the important concepts across. So there's our pathogen, there's the antigen whose story we are going to follow here. Okay, right. So now what I'm going to do is I'm going to draw the nucleus again, and again it's going to be a story that's going to involve the rough endoplasmic reticulum, so I'm going to draw the rough endoplasmic reticulum out here, like so. Okay, right. So this pathway is going to be slightly different. It's not going to follow the same story as we saw for the case of an intracellular pathogen. So the way we're actually going to chop up these antigens and get fragments is different in this case. So we're not going to actually use the proteasome in this case. What's going to happen is we're just going to fuse the lysosomes with the phagosome. That will break down the antigen into fragments, and that will do the job enough. So we're not going to use the proteasome in this case. So we're just going to form a phagolysosome, like so. So I'll just draw the lysosomes coming in. So here in blue, these are the lysosomes coming in to fuse with the phagosome, and that will now result in this antigen being broken down into fragments. Okay, so here I'm just now going to draw the antigen fragment. Of course, you'll get a lot more fragments of this pathogen following the fusion of the phagosome with these lysosomes and the formation of the phagolysosome, but they're not particularly relevant for our story. So just to keep it nice and simple, here is the fragment that we're going to follow that has been created from this antigen when the lysosomes fused with the phagolysosome. And for this pathway, it's going to actually have to be between 12 and 20 amino acids long. It's also going to have other characteristics, but we'll come on to those later because that's more complicated. For now, just know that the antigen fragment is going to need to be between 12 and 20 amino acids to be presented on MHC class 2 molecule. So this that we've got here, this is the phagolysosome. Okay, right. So now leave the phagolysosome aside and come back to the rough endoplasmic reticulum. So we are going to be synthesizing MHC class 2 protein complexes, and these are going to end up in the membrane of the rough endoplasmic reticulum, and I'll draw one here. So this is not MHC class 1, this is now major histocompatibility complex class 2. So this is MHC class 2, so I'll put it in a violently different colour to MHC class 1. So we'll have MHC class 2 now in yellow. It is a different protein complex, and we'll come on to the structures of MHC class 1 and MHC class 2 later on. Okay, so here is the MHC class 2 protein complex in the rough endoplasmic reticulum. Now, remember in the previous story we brought the fragment to the MHC class 2 complex. In this story we're going to bring the MHC class 2 complex to the fragment. Now, what has to happen in the rough endoplasmic reticulum before you start moving the MHC class 2 towards the antigen fragment here is you have to put something in its peptide binding groove to fill that peptide binding groove to make sure that other things don't bind there. So this has a great capability of binding to peptide fragments. You don't want peptide fragments to go in there until we actually get into the phagolysosome, and then the peptide fragments from the antigens of the pathogen can go in there. So what you actually stick in is something known as the invariant chain, which is just going to sit in there until we actually get into the phagolysosome. So this here, this is representing something known as the invariant chain, which is just to keep the peptide binding groove free from little peptides until we get into the phagolysosome. So there in blue, that's the invariant chain. Now what's going to happen is we're going to move the MHC class 2 with its invariant chain to the phagolysosome, and the way this is going to work is exactly what we've just seen. So it's going to have to go through the Golgi, then into a secretory vesicle, and that secretory vesicle can then fuse with the phagolysosome. So it comes through the Golgi, and I'll just draw 4 cisterna this time for the Golgi here. So it comes through the Golgi into the trans Golgi, just like what we've drawn here. Sorry, into the cis Golgi first, then to the trans Golgi, then into a secretory vesicle here, like so. I'll just draw it in here. So here's the MHC class 2, here's its invariant chain. In yellow there's the MHC class 2, and then in blue the invariant chain. And then what's going to happen is this secretory vesicle is then going to fuse with the phagolysosome here, and we're then going to deliver the MHC class 2 with its invariant chain into the phagolysosome. Now what happens when we get into the phagolysosome is that the invariant chain moves out of the MHC class 2 peptide binding groove and is also going to be broken down by some of the enzymes in the phagolysosome. What will then replace the invariant chain is one of these fragments from our antigens of the pathogen, and this is the fragment that we're following, so we'll have this one sitting now in that peptide binding groove of MHC class 2. So now here in yellow, here's the MHC class 2. So that's the way that we get these antigen fragments from our pathogen actually loaded onto major histocompatibility class 2 complexes when we're dealing with an extracellular pathogen. And then what will, of course, will happen is this will fuse with the plasma membrane so that these MHC class 2 with peptide complexes are put on the surface of the antigen presenting cell. So here is the antigen fragment, and here is the MHC class 2. Okay, right. So to summarize what has happened here, when we're dealing with an extracellular pathogen, that extracellular pathogen will be phagocytosed by the antigen presenting cell. It will be broken down by fusing these lysosomes with the phagosome to form a phagolysosome here. Therefore, antigens of this pathogen will be broken down into loads of little fragments. Now what's going to happen is we're going to deliver MHC class 2 complexes into the phagolysosome, and I've drawn one MHC class 2 complex. You'll actually be bringing loads of the things. So the MHC class 2 complexes, they begin their life in the rough endoplasmic reticulum where they have become integral membrane proteins. So this is where we built the MHC class 2 and made sure that it was integrating into the membrane correctly. And to prevent anything binding in its peptide binding groove too early, we put in that invariant chain. It's then transported through the Golgi apparatus into a secretory vesicle, and this secretory vesicle, which will contain loads of MHC class 2 and invariant chain complexes, will then fuse with the phagolysosome. The invariant chains will come out of these MHC class 2 complexes, and then fragments of the antigens that are between 12 and 20 amino acids. So this is an important difference between MHC class 1 and MHC class 2. MHC class 1 likes to present fragments, its peptide binding groove is big enough to have peptide fragments between 8 and 10 amino acids. MHC class 2 has a bigger peptide binding groove, and this can take peptide fragments between 12 and 20 amino acids, so longer peptide fragments in it. What will now happen is, once we've got these peptide fragments bound in the peptide binding grooves of the MHC class 2 proteins, the phagolysosome will fuse with the plasma membrane and will now have loads of MHC class 2 complexes with peptide fragments on the surface of the antigen presenting cell. So where we have got to by the end of this video then now is we have seen that if we have an intracellular pathogen, its antigen fragments will be presented on the surface of antigen-presenting cells in MHC class 1 molecules. Whereas if we've got an extracellular pathogen, its antigen fragments will be presented on the surface of an antigen-presenting cell in the company of MHC class 2 complexes. This is how we're going to initiate these two different parts of the adaptive immune response. MHC class 1 with peptide fragments will initiate cell-mediated adaptive immune responses, and MHC class 2 with antigen peptide fragments will initiate humoral adaptive immune responses. Right, so we're going to have a break here. In the next video, what I want to do is discuss MHC class 1 and MHC class 2 in a lot more detail. I want to discuss what conditions the peptide fragment has to actually have to be able to bind in the peptide binding groove, because MHC class 1 and MHC class 2 complexes cannot just bind any old fragment between their required length of amino acids. There is some requirement on the peptide fragment, and I want to discuss that. I also want to discuss the genetics of MHC class 1 and class 2 molecules, because it's complicated and it is important to understand it, because if you're reading research papers on the autoimmune diseases, for instance, you will all the time hear about the different alleles of the different MHC class 1 protein complexes, for example, being associated with this disease. Okay, so we'll do that in the next video.